Sanger DNA sequencing is a chain termination method used to determine the sequence of DNA. It is a groundbreaking technique for determining the nucleotide sequence of DNA. Now, question how it works. DNA polymerase incorporates chain-terminating dideoxynucleotides, ddNTPs, during DNA replication in vitro. The ddNTPs lack a 3' OH group, which prevents the formation of a phosphodiester bond between nucleotides and terminates DNA strand elongation. This results in DNA fragments of varying lengths. How to read these DNA fragments? The answer is: the DNA sample is divided into 4 sequencing reactions, each containing a standard deoxynucleotide and DNA polymerase. A single ddNTP is added to each reaction, and the newly synthesized DNA fragments are separated by size using gel electrophoresis. It was used in the Human Genome Project to determine the sequences of human DNA fragments. The chain termination method is more efficient than other methods, using fewer toxic chemicals and less radioactivity.